Hi, so my name is Jessica Perkins and I chose to do my presentation on how paternity tests work. So have you ever wondered how paternity tests work, or perhaps has someone ever asked you, who's your daddy? So what are paternity tests? So when a baby is conceived, each parent contributes half of their DNA or genes, which is the heritable information, to their offspring. Occasionally, a mother may be unsure who the father to her child is, and this is when DNA paternity tests come into play. Paternity tests rely on genetic profiles from the child and the father being tested. DNA paternity tests are highly accurate scientific tests that prove or disprove the assumed biological father of the child. The reasons paternity tests are used. Paternity tests are used for many reasons, including personal, medical, and legal reasons. So home tests are typically done for personal reasons because they can't be held up to like court standards or anything. So a few of those reasons for home tests are for peace of mind for the family. If they are unsure that the assumed dad is actually the biological father, and they are also used for adopted children if they are invested in finding their birth parents. And adopted children can also need paternity tests for medical reasons if they have medical issues going on. So paternity tests are also done for medical history to help doctors take preventative action for their patients. The last leading cause for the use of paternity tests are for legal reasons. They may have to decide in court child support or custody arrangements if a mother and a father decide to no longer be together, to be separated or be divorced. How do paternity tests work? So analyzing DNA is the focus of paternity tests. The trillions of cells that are in our bodies all contain a nucleus that hold our DNA. There are cells in and on every part of our bodies, and therefore DNA can be sourced from many easily accessible areas, including our skin cells, our saliva, and our hair roots. They all contain DNA. The samples collected are examined by scientists who look for similarities in the genetic makeup of the father and the child. And a deeper dive on how paternity tests work. So DNA is the molecule of inheritance, what you inherit from your mother and father. So DNA is made out of only 4 nucleotides, and they form double helices. During DNA replication, the paternal DNA separates and forms 2 new DNA helices, with 1 paternal strand each and 1 new daughter strand each. This replication is called semi-conservative replication and can aid in matching a child to a suspected father by matching half the alleles of the tested offspring to the suspected father's alleles or markers. Testing and procedures. So paternity testing requires an uncontaminated sample of DNA. Home testing, such as adenosine, is able to gather a large enough sample of DNA to be used in paternity tests, where it must be put through a process to become uncontaminated. The most common paternity test is with an affordable kit that you can buy at your local pharmacy. And the process of the home kit involves swabbing the inside of the father and child's cheeks to collect saliva and cheek cells. And the cells contain DNA, but they must be uncontaminated and put into a pure form to be able to be used in a paternity test. Testing and technology involved. So a chemical process called lysing is used to break open the cells collected. This process separates the DNA from the nucleus. And then robotics are then used to separate the DNA from the remaining cell parts through another chemical process where the DNA is preserved on beads and the other cell parts are then washed away. Elution is the last step where a robot collects the DNA from the beads. So they collect the pure form of the DNA from the beads. And then the DNA is now ready to be used in a paternity test because it is now in a pure form and uncontaminated. Testing and technology involved. Scientists place the collected pure DNA into a special primer solution. And then this solution finds and makes copies of the targeted DNA needed for the paternity test. Heat then separates the double-stranded DNA, and as it cools, the primers bind to the single strands of DNA and make two copies of DNA. This process is then repeated 28 times and makes millions of copies of the DNA. The DNA is then viewed by a machine called a genetic analyzer. This process is referred to as polymerase chain reaction, or PCR. And the PCR makes 16 to 18 genetic copies called markers to form a DNA profile of the child and the father or parent. So the DNA collected for the paternity test is different lengths and sizes. And then they use software and it measures the DNA represented by two alleles to read the paternity test. Economic impacts. As you can imagine, raising a child costs money. So a negative paternity test could potentially save a suspected father from providing income for the child and supporting them if they are not the father. A positive paternity test could potentially cost a suspected father a substantial amount of money and support throughout the child's life. Scientific impacts. So inheritance is the process by which the traits of an organisms are passed to their offspring. Homologous chromosomes are the chromosomes inherited from the parents and are within our DNA. In the mid-1800s, Gregor Mendel experimented with pea plants, and he discovered that the pattern of inheritance during sexual reproduction. The best way to learn about inheritance today is still to examine Mendel's experiments. He was way ahead of his time. So in conclusion, I believe scientists will continue to make enhancements and findings with DNA and ways to test paternity. So when a child shares half of their markers with the potential father or mother, then a paternity test will determine they are the parent to the child. When the paternity test shows the markers do not match, then the person will be excluded as a potential parent. And DNA tests are very accurate. They're actually 99.99% or greater accuracy. And these are my references. And continued. Thank you for watching.